Let's know more about chromosomes, DNA replication, and mitosis together. In this class, we're gonna simply help you understand the structure of chromosome and quite other concepts. Okay, let's start with the structure of chromosome. As you can see, we have the chromatid, which is this, a single shape of that, and another one which is exactly identical to the first chromatid here, and these two are linked by a centromere. Okay, so this is actually called a centromere. Okay, highlight that. This is a chromatid, another chromatid, and the centromere. All this structure is called a chromosome. It's made from two sister chromatids. The reason we call them sister is because they are identical. Attached by centromere, that's what we've just said, made of two DNA molecules. Actually, you might have thought, like, these are too many. Like, doctor, these are like millions of DNA molecules. Oh, actually not. These are only two. This has only one DNA molecule, and this contains the other. The reason why they look many is because they are condensed DNA. And that's what we're going to discuss later on this class. We know that we have interphase, which is the phase we prepare for mitosis that happens before it. At this phase, we have chromatin. Remember when we've discussed together the organelles of an animal cell, where we have a nucleolus, some chromatin, and the cell, the nuclear membrane, and the nuclear pores here. This was the chromatin we're talking about. Do you remember that? This is the chromatin. It's loose, it's uncondensed, it's unfolded DNA. And then it gets replicated, it doubles. We've always said that it doubles, and this is called semi-conservative replication. Semi-conservative replication. Let's just write it down another time. Semi-conservative replication. Okay, that's the phase where they get doubled. And of course, we know that we double it in order to have the number of chromosomes identical, or not only identical, I mean the same for every member of the species. Okay, I mean for humans we have 46, so they have to be doubled. Okay, to be 92, so that when we divide them once more, we have 46 and 46. Okay? It's like they're not going to be 23 and 23. We're not like fleas. I don't know what. Okay, that's the chromatid. Another chromatid, centromere, and this is all a chromosome. We talked about the interphase, the semi-conservative replication, semi-conservative replication. Okay. And basically, the chromatin and the chromosome are the same. They're just DNA. But the difference is this is unfolded and uncondensed. That's why we find it loose, like that. So this is unfolded and uncondensed DNA, while the chromosome has folded and condensed DNA. We can find the chromatin structure through the interphase, while we can find the chromosome through mitosis. Okay, so this is through mitosis and this is through interphase. Okay, again, I know this name is quite confusing or a bit difficult for you to memorize it. It is again called the semi-conservative replication. Okay, now let's know more about the cell cycle. They are sequences of events taking place between cell divisions and the next. What's going on? Okay, the three phases are, of course, interphase, which we've talked about, and about the semi-conservative replication. Also, mitosis, of course, it's quite famous, and then cytokinesis. Cytokinesis, I don't know if I'm pronouncing it correctly, excuse me. I think it's just cytokenesis. Exactly. Yeah. For interphase, it actually has three main phases, which I call G1, G2, and S phase. Their order is actually starting from G1, then S phase, and then G2, the third one. Okay, for G1, it's the first growth phase, where the organelles are being copied. Why, doctor, are we copying them? It's because you're actually gonna divide them. We just said there are 46 chromosomes divided, 92 divided, and dividing everything, not only the chromosomes. There's gonna be a new whole cell with all of its organelles, cytoplasm, vacuole, chloroplast if it's a plant cell, and all that stuff. Even mitochondria, all of these have to be copied. Organelles being copied, protein synthesis, increase in amount of cytoplasm in order to divide it. Like, you're not gonna divide the same amount of cytoplasm. This is not gonna be an appropriate or a normal cell, okay? It's gonna be abnormal at this time. So again, in the first growth phase, we have the organelles being copied, protein synthesis, and the amount of cytoplasm is increased. Okay, for the second phase, which is the S phase, the synthesis phase, where the DNA replicate by semi-conservative replication, and this process produces two identical copies of DNA molecules to form chromatids. Okay, semi-conservative replication, it produces two identical copies of DNA molecules to form chromatids. Until now, we still have chromatids. We didn't talk about any mitosis, okay? Chromatids now, where the DNA replicates, it's synthesized, semi-conservative replication, where we have two identical. Take care of this word, it's a key word. Whatever exam you're taking, they are identical copies of DNA molecules to form chromatids. Okay, and then we have G2, which is the second growth phase continuing to this. Okay, enlargement of the cytoplasm. So here the amount is increased, and here's enlargement. The cell continues to grow. Enlargement of cytoplasm, the cell continues to grow, and new DNA molecules are checked for errors and usually repaired. So not only are they checked for their errors, they are also repaired. So again, what happens here? Point number one, enlargement of the cytoplasm. Then the cell continues to grow. New DNA molecules are checked. We check them and usually repair them for their errors. Okay, here's a question that's repeatedly present in your questions, also in the exams and so on. Why mitochondria replicate during early stages of cell cycle? I mean, we have to. Like, how's gonna cell survive without mitochondria? Can you understand? As the number of mitochondria will later be halved by the end of cell divisions to provide energy for mitosis or growth. So here we provided not only the reason, like we replicated, this is the reason. It's gonna be halved and we need it. And the reason why, like, why am I gonna need mitochondria anyway? Like, I know it's not gonna be present in the cell that's divided, okay, if we divide them into two cells. But why do I actually need it? That is the reason. To provide energy for mitosis and growth. We're actually doing mitosis. Am I doing mitosis to provide energy for mitosis? Yes. This is how we live, okay? So as the number of mitochondria will later be halved by the end of cell divisions, and to provide energy for mitosis and growth. Okay, here we come to mitosis, the major topic or the major concept of today's class. Okay, mitosis is what we actually need interphase for. Like, if there wasn't mitosis, there wouldn't be even interphase. I know, let's not get into these details, but mitosis has 4 main phases, which are prophase, metaphase, anaphase, and telophase. Okay, let's get through them. At prophase, this is the very first phase of mitosis, where the chromatin, which we consider to be inside like a normal nucleus, this chromatin is now condensed into chromosomes, where each chromosome is made from 2 sister chromatids, the same thing we've been discussing, attached by a centromere just in between them. Like, I don't know how to draw. Nuclear envelope breaks into vesicles. So we actually had a normal nucleus like that. This was normal and it has nothing wrong with it. Nothing's missing. Has a nucleolus and the chromatin and the nuclear pores and everything. And now we said that the chromatin is now condensed. The nuclear envelope breaks into vesicles. Take care. It does not only break, but breaks into vesicles. Okay, vesicles. The nucleolus disappears. The centrioles start moving towards poles of the cell. These are called the poles of the cell, and these are also like these centrioles. If they want, I didn't declare centrioles before. Okay, these are the centrioles that are made of 9 triplets of microtubules. Okay. These are the centrioles, and they move to the poles of the cell, right here. So we said now that the chromatin condenses, the nucleolus disappears, the nuclear envelope breaks down into vesicles, and the centrioles start moving towards poles of the cell. The spindle fibers start to form. Okay, these spindle fibers that help break the cell into 2. Okay, these are called the spindle fibers. Like, we're not gonna talk about its details and its function right now. We're focusing more into mitosis. Okay, the spindle fibers right here start to form. This was about prophase. What then happens in metaphase? The centrioles, which have just moved towards the poles, are gonna reach it. Okay, we were just saying that it's gonna move. It now reached the poles of the cell, and the spindle fibers are fully formed. They attach to the chromosomes by the centromere at kinete core. This is the position, the exact position in the centrosome. It's called this name. Okay, I don't really suggest you should know it, but okay. Attached to the chromosomes by the centromere. Okay, so at metaphase, which is the second phase, again, I'm repeating so that you guys can find it easier to memorize and elaborate to the examples I'm saying and everything. Okay, so again, the spindle fibers are fully formed after the centrioles have just reached the poles, and the spindle fibers start to attach to the chromosomes at their centromere, okay, at kinete core. They then align or line up at the chromosomes at the cell equator. So again, speaking of the positions of the cell, this region is called the cell equator, and these are the cell poles. Okay, then let's move to anaphase, the third phase, where the centromere divides. It does not break, it divides. There's a part that's left here and there's a part that's left here. Okay, so it divides. The centromere divides. The spindle fibers shorten. Okay, this is the key word. They shorten and the centromeres divide. The spindle fibers are gonna pull the chromatids apart at the centromere. It's gonna pull them at the centromere. At their centromere, they're gonna be pulled, you know. The sister chromatids are gonna move to the poles of the cell. They were aligning here originally, okay? Then they're starting to move apart of the cell equator the chromatids now become single chromosome. They are actually chromosome, but they're single, okay? Then we reach the fourth and final phase of mitosis, which is the telophase. We actually need a nucleus for every cell that exists, like almost every cell, okay? So we couldn't divide them if there wasn't a nucleolus, a nuclear membrane, and all these cell organelles. So the nuclear envelope preforms. I want you to understand it, not just like memorize these steps. Like it's the final phase. We're gonna prepare to the separation to be like a mature cell. I don't mean like mature, I mean like a full cell, okay? The nuclear envelope preforms and the nucleolus, reappears. All of a sudden, I reappear again, okay? The spindle fibers break down. I no longer need them. They were used like to push and pull the chromatids and everything. They're now like useless. I'm not going to need them, so they're going to be broken down, and the chromosomes are uncondensed and unfolded into chromatin. We started to the first point we've talked about, and the cytoplasm constricts. By constrict, I mean like they form this shape. It's now getting ready to be separated, okay, and becoming like two cells. Cytokenesis. Okay, this is the correct pronunciation, I know it now. Cytokenesis is the division of the cytoplasm. If you can observe how the cytoplasm starts with cyto, and the cytokinesis also starts with cyto. This means like it refers to the cytoplasm. It has something to do with that, okay? Division of the cytoplasm and formation of two genetically identical daughter cells with the same number of chromosomes. This is simply the separation, okay? It has two types, the animal cells and the plant cells. For animal cells, it's the breakdown of remains of spindle fibers. That's what we've said here. The spindle fibers are going to break down, and the cytoplasm constricts, constriction of the cytoplasm, and formation of cleavage furrow. What is this cleavage furrow? It is this. This is the cleavage furrow, okay? You're just going to know it by name. Okay, the cleavage furrow is formed. A ring of contractile fiber tighten around the center. It's like something it's holding very hardly on it that's gonna be broken down out of the pressure pressed on it. You get me? So this is called the contractile ring. Again, because these names are new and I know you're gonna like get lost in that. We have a contractile ring. Okay, and a cleavage furrow. Contractile ring and a cleavage furrow, where a ring of contractile fiber tighten around the center of the cell until two cells are formed. It's also like it's the contractile fiber too. Okay, so we just said this here like a recap. Contractile ring tighten at the center. It tightens at the two cells at the center, and it increases constriction cytoplasm. Formation of cleavage furrow continue till two cells separate, and we finally have two cells. This happens all the time, okay, in your body. Now we've come to the part of cytokinesis in plant cell. Okay, it's quite different, where the spindle fibers direct the Golgi vesicle containing cell wall material and enzymes to the cell equator. You remember what was the cell equator? Was the center, like the center. If the cell was round, this is the cell equator, okay? So the spindle fibers direct the Golgi vesicles containing cell wall material and enzymes to the cell equator. The vesicles are going to enlarge and fuse together, forming cell plates perforated with a plasmodesma. Division in cytoplasm. Okay, you're gonna know this like more clearly right here. Sorry, I want to highlight this part. Like you guys have to read this. diagram. Let's just take a quick look on it. I want you to see it well, I know. We have that some spindle fibers remain and guide the Golgi vesicles to the equator of the cell. These vesicles are gonna enlarge and then fuse all together, forming a cell plate, which is then called the plasmodesma, okay? The cell surface membrane that's formed, okay? And we have these plasmodesmas here. This is the basic structure. The cell wall forms within each vesicle. Each vesicle that the spindle fibers are going to guide, this is eventually what it's going to be. Okay, the small gaps are the plasmodesma, what this formed, okay? So the spindle fibers direct the Golgi vesicles containing cell wall material and enzymes to the cell equator. And those vesicles enlarge and fuse together, forming cell plate perforated with plasmodesmata. Okay, so they form cell plate that is perforated with plasmodesma, or you can also call it aka plasmodesmata. Okay, and of course the division in the cytoplasm, the cytokenesis, division of cytoplasm. An implant cell, which is special about it, that the spindle fibers are gonna direct the Golgi vesicles to the cell equator, which of course is carrying with it some very special cell wall material and enzymes to the cell equator in order to form this. Okay, it has the enzymes and the cell wall material for that specifically, okay? And of course the division in cytoplasm. Okay, why do we do this cell cycle like always, all the time? We have either interphase or mitosis or cytokenesis going on in our cells. Why? Like, what is its function, its significance? Okay, they control the growth of an organism, they repair its damaged tissues, replace its dead cells, increase the number of cells due to cell divisions, and all these functions. Like, of course, we wouldn't exist if it wasn't for that. We wouldn't grow. We'd be still young. And there's our damaged tissues. If you got a bruise, if you got hurt, it's never gonna be well anymore. Like, you're gonna stay forever like that. You may bleed, there's no repair, there's nothing going on. Okay? Can control the growth, okay? Repair damaged cells, replace the dead cell, increase number of cells due to cell divisions. And of course, it increases in size of cells during G1, G2 phases. Asexual reproduction, we're going to talk about this later, no need now. Replication of DNA and new cell organelles and formation of new cytoplasm. The cell cycle is to replicate the DNA, of course. It's like main something. New cell organelles and formation of new cytoplasm to produce genetically identical cells. Like, that's a must. Specialization of cells take place after cell division. Like stem cells, you know them? They're specialized just by mitosis, and they undergo it. What is the significance of mitosis like that? There are some points that are here too. Of course, the growth of an organism, giving rise to genetically identical cells with nucleus having a full set of chromosomes. It's not missing. As we said today, 92. When we divide them, we get 46. It's the same human species. Or else we're gonna turn into fleas or fish or whatever, okay? Replacement of damaged dead cells, regeneration of tissue or an organ. So about the cells, they are replaced. About the damaged tissues, they're repaired. And we regenerate even more tissues or organs. That's why, or that's how you grow. Okay, and it's of course used in asexual reproduction or cloning of the chromosomes and so on, okay? Thank you for watching, everybody. I thought this session was pretty beneficial. Not for only you, but for me. I loved it. Thank you, everybody, for watching.